Assalamualaikum to everybody. Welcome to today's breakfast talk. Today we have for our speaker, it's Dr. Rozaida Po, and I am Suzita from the Biomedical Science Department. Both of us are from the Biomedical Science Department. Dr. Rozaida is a senior lecturer at the Biomedical Science Department, as well as our Biomedical Science Program Coordinator. Her her current research investigates how a medicinal mushroom extract affects the brain after injury, focusing on the role in reducing inflammation, protecting brain cells, and supporting repair. And today she is going to share with us on how traumatic brain injury can be studied in zebrafish models. And she'll take it away now. Thank you.
Thank you, Madam Moderator, for the kind introduction. Assalamualaikum and a very good morning to everyone. Thank you for joining. And today I'll be talking about the role of the zebrafish in traumatic brain injury research. TBI models are actually scalable, reproducible, and also pathophysiologically relevant to human TBI. But I'll also be highlighting a bit at the end why, when we say scalability, it does not necessarily mean simplicity, especially when we are working with some models that I'll mention later, like the telencephalon stab injury and also the modified Mamoru weight drop. So moving on, there are some challenges in studying traumatic brain injury. We know that the human brain has the complexity of the brain injury that occurs in the human brain is there. There are heterogeneous injuries that can occur in terms of severity, the causes, age-wise, what was the age that this injury occurred, and also in terms of any existing comorbidity that might be further exacerbating whatever is being inflicted, right? So there are also, at the same time, limited treatment options. And for this part here, right, in terms of the limited treatment options, to date, despite decades of research, few therapies improve long-term outcomes beyond acute stabilization. And this could be partly also due to the inadequate models that are being used. So in terms of functional recovery, there are difficulties in measuring functional recoveries in terms of cognition, motor skills, and also memory. And this is not to put down the rodent models, but it is more to introduce why we are looking at the zebrafish model, because relatively rodent models would be slower in terms of its throughput, and also, of course, it is more costly. So as you can see here on the right side, right, there is this very simple diagram, which is just showing how the different types of injuries are occurring in the brain, right? So when upon infliction of TBI, right, there will be this primary injury that causes stretching. Let me just, okay, stretching, compression, as well as tearing of tissues, right? And from there, this will lead to secondary injury. And this is, the secondary injury is actually a response to the primary injury. And this results in certain cellular events, molecular events that can evolve over time. And this evolution over time are different at different stages, and that makes it more challenging in terms of research, right? Okay, so moving on. So why do we then use adult zebrafish for TBI? So I would like to make a disclaimer from the start that the title was Zebrafish Models in Focus, but I would be focusing more on the adult zebrafish per se. And again, this is not to do this kind of a comparison that my horse is better than your horse kind of thing, but it is more about the focus, because I would not be covering the larval zebrafish or the embryos, right? And each of those other models have their own place in terms of TBI research as well, right? So with that, adult zebrafish, they have these fully developed mammal-like brain structures. And the brain anatomy are more relevant to the human TBI pathology. So when I say here more relevant, probably this would be in comparison with the larvae, for example, right? And these include the white and gray matter structures. Additionally, there are more realistic injury modeling that mimics, for example, the closed head injuries in humans. And what makes the zebrafish in general, not necessarily adult, more unique in terms of TBI studies is that zebrafish have these regenerative capacities, and this sets it apart from, for example, rodents, right? So therefore, when you do TBI on these zebrafish, there will be the neurons which are damaged, and we can study how and how fast they can be regenerated. In addition, there are similarities in certain genetic and molecular pathways with humans, more than some other models, and that makes zebrafish attractive. In fact, I've attended this responsible care and use of lab animals previously, and we have been given this lecture on the fact that zebrafish has all these wonderful attributes, which I'm not going to expound too much at length, but suffice to say, these similarities in terms of the molecular pathways, for example, helps to clarify certain biological consequences and probably pave the way for therapeutic opportunities as well. But that is not to say that these are going to be then directly translated from tank to bedside. Okay, so that would be another disclaimer. So it's more about understanding certain biological consequences and perhaps harness that for future therapeutic purposes. And of course, zebrafish, if you compare with rodents, they have a higher throughput and they are more cost effective. And this is related to the zebrafish in terms of the history of it, right? Why zebrafish? Why not some other fish, right? We have so many other fish in the world, of course. So it has been studied, and there is a very interesting history about it. I would really recommend you to go through that. And maybe a summary of that would be, it was found that these zebrafish can breed very rapidly, right? And also, they are quite hardy. That means they can stand the lab conditions better than certain other fish. So that makes them more attractive to be used in the lab. And so the high throughput and cost effectiveness of it comes as a consequence of that, because they are rapidly, they can breed rapidly, okay? So in terms of behavioral assays, they can mimic certain behavioral landmarks when you compare with the humans. And also there are certain ethical considerations, and I think those are quite a number of adult characteristics that make these adult zebrafish very attractive for research in general, and in particular here, TBI. So with that, let me move on. And there are some of the more common models, I would say, but these are not limiting to these three that I'm showing here, okay? Definitely not. And again, because I'm emphasizing on the adult zebrafish, right? So of course, there are those larvae models that can also be used for TBI studies. Definitely, they have their own place, okay? They could have their own cons, but there are certainly also pros for them. So let's look at the one model, which is a blunt force TBI. And you can have a variance of this, but one of it that I would like to highlight, which we are doing in our lab, and that's why I'm highlighting, is the modified Mamru weight drop. And this particular type of TBI model, it induces blunt brain injury. And this was originally adapted from the rodent model. It delivers calibrated mild, moderate, or severe injury. And these are reproducible and scalable, as mentioned earlier in the common slide, but it is even more reproducible compared to the brain stab here, as you can see. And so it has its own place again, right, compared to the other models. And what makes it special is that it can recapitulate human TBI hallmarks, and we have seen seizures and also hemorrhages in the fish that have been subjected to severe, especially severe TBI using this method. Okay, so it's very clear. And for another model is actually the PHIFU, this is short for closed head pulsed high intensity focused ultrasound. We did not do this particularly in the lab, but this particular model will cause mechanical TBI. And this type of model enables dose-dependent injury, downstream evaluation of locomotor deficits, anxiety, social behaviors, and responses to pharmacological intervention. And these are used even for other models like PTE, post-traumatic epilepsy. And the other model that we are doing in the lab right now as well is the brain stab injury, whereby the penetrative injury is being induced specifically on the zebrafish telencephalon. It can be done on the optic tectum as well. And between these two, there are also their own pros and cons. It relates to the part of the brain when you're talking about telencephalon, it will be more related to the cognitive and memory part of the properties. And so if you want to study those particular characteristics, then you would go for the telencephalon. But if you were to study perhaps the visual or sensory motor capabilities following TBI, then you maybe want to look at optic tectum. And of course, in terms of the anatomy, telencephalon is slightly more challenging to reach to, and optic tectum is more easier. It is more superficial dorsally, and so they have their own pros and cons, right? So, and this brain stab particularly is used to explore neuronal regeneration and migration. So it is more local, focal, and it is maybe mainly used to study maybe the regenerative properties rather than the blunt force type, which would want to look maybe at certain seizures, properties and things like that, right? And hemorrhage. So moving on. So here, this is the real picture of a zebrafish, if I may point it out to you, from the lab. And so as a comparison here between the two methods which we are currently doing in our lab, right? So we call it complementary because of the nature of what I have mentioned earlier in the previous slide. So to recap, the telencephalon stab injury is what we are doing specifically. So we are looking at the cognition and the memory part of it. And the nature of it is that the injury is localized. It will model the glial responses, right? The regenerative as well as the radial glial cells responses. And of course, this requires surgery compared to the weight drop method, because weight drop itself implies that you are dropping a weight onto the head of the fish, whereas for the stab injury, you're stabbing something there, right, which is a telencephalon. So from the nature of it itself, it is relatively slower, but it is more detailed perhaps in terms of certain objectives that you want to achieve, right? And so by comparison, the weight drop method is faster and scalable, and in fact, it's cost-effective, more cost-effective as well. So that's what we're doing in the lab. And just as a snapshot to make it clearer on what I was talking about and what we were doing in the lab. So this is depicting the telencephalic stab injury, right? And you can see there's a tiny red cross there, which indicates a position of vertically induced injury, right? And this pentagon here is indicating like the anatomical landmark to guide to the site of the lesion. So the labeling of this zebrafish brain itself will zoom into this part here. We are actually looking at this part here, the dorsal telencephalon, right? And we are poking usually on the left hemisphere, okay, of this telencephalon. So that's what we are doing. And that's the student who's doing it, Azwin. And for another model, which is the weight drop model, this is another student. And it is called modified because the original method is actually derived from the rodent model. So that would be the original one done back in 1994. But here the modified one is a method that is being done on the zebrafish. And so there would be slightly different approaches to that. And if you can look at A here, this is the dimension and condition of the setup to induce blunt TBI in adult zebrafish. And there are metal balls of different weights to inflate the varying severities of TBI. So if it's 1 gram, 2 gram, 3 grams, this corresponds to mild, moderate, and severe TBI, right? And when we subject the zebrafish to the severe TBI, that's what we can see that there are very marked seizure-like movements, okay, compared to compared to those who were subjected to mild TBI, okay? So this is actually B, which is a close-up showing the position of the anesthetized zebrafish in the clay mold, right? And there's a metal disc that we place on top of that zebrafish, okay, to ensure that the TBI is being diffused, right? And the aluminium tube is going to be positioned precisely above the head. And you drop the ball and you bang the head, right? And so for C here is again showing the area of impact, which is different from the stab injury, whereby we're looking at the telencephalon. But here you see that it is between the optic tectum and the cerebellum, right? So this ensures that injury is diffused across the brain after impact. So moving on, there is one aspect of this whole study that we almost did not realize until quite some time at the end, is that randomization has to be done in terms of when we want to sample the fish each time for the different time points. And so this is just a cartoon that I have sourced from this particular website. So to understand the basis of that randomization, this could be a go-to website that you can start with if you're interested, right? If you're so inclined. So it just implies that although they have a random number generator, but when everything came out as 9 each time, when you draw 6 times, you get 9 each time. There's a question about, is that actually random, right? But I'm just maybe putting across a point that randomization is something that we would want to look into, okay? So, and for example, here, if I may show our scheme on attempt at randomization after several back and forth with the consultation on how best to randomize our sampling. So what we have for every treatment, if there were to be 4 treatments, right? This is a scheme. So maybe for your purposes, you could have a different scheme. But if you have 4 groups, right? And the group 1, let's say, could be just the injury without the treatment. And then group 2 is the one that has the base material in the mushroom extract that we have, right? And then the third group could be the low dose group, and maybe group 4 is the high dose. So we would want to have there are going to be 3 time points that we are going to look at the fish brain, which is at 1 day post-injury, and 3 days post-injury, and also 5 days post-injury. So after we injure them with a stab wound, we're going to wait for 1 day, and we're going to take 2 out of this tank, which contains 6 fish, right? And because we need 6 altogether for each time point, we're going to take 2 more from tank B, and we're going to take 2 more from tank C. So this part here was the part where we almost did not realize, until we consulted with the statistician. So we were going to take maybe all from 2 tanks only. For example, we thought we'll just divide into 2 tanks. We're going to take two. And so that is one form of random sampling that we can get away with. So if I were to go through here, because the sampling is destructive, you want to select 2 fish. And why 2? Initially, we wanted to take 1. We thought we can get away with 5 per group, right, in a tank, because statistically, in fact, minimum would be 4 for our purpose. But we wanted to then maybe have a little bit more. So we put in 5 because of the biological variation and also what can happen to the fish down the line. Perhaps one of them might, die or things like that. So we wanted to have a backup of 1 more fish. But then if we have 5 per tank, that means you want to take, if you wanted to take 2 each time, we would have the last fish being alone in the tank up to 5 days post-injury, right? And that's the thing. When I mentioned just now about the RCU lag is very important because the RCU lag was short for the course on the responsible care and use of lab animal, right? So that kind of background knowledge is important because then we know that these fish, and in particular these zebrafish, they are social animals. And when you isolate them, you have the last one waiting in the tank until the 5th day post-injury, then these can actually alter the physiological markers like cortisol and oxidative stress indicators. And we are actually looking at oxidative stress indicators for this particular setup. So you don't, basically when you're talking about experimentation, you want to control all the possible factors that you can. So this is where you want to design it in such a way that all these other bias, potential sources of bias and variations can be minimized, right? So with that, I'll move on. And so just about what's been found so far, but from other studies, right? Because we are going to give the mushroom. Right now, the student is in the middle, in the thick of doing her stabbing of those. If you can look at the previous slide here, it's going to be like 18 fish per day, like 2, 2, 2. But when she wants to start this whole set, right, it has to be stabbed all 18. Then from there, you sample at 1 dpi, and then at 3 dpi, and then at 5 dpi, right? So with all the 18, sometimes she felt that it was a kind of extreme sport, but I'm sure she will get the hang of it. And so we have not found as yet for our own study, but I would like to maybe highlight for other studies, right? And for this part, I'd like to just refer to, so if I may go back to that sampling just now as well, this also means that if you just take from a particular tank, right, and not take from all 3 tanks for representing the 3 time points, you might have what we call the tank effect as well. So that's something that you can consider for zebrafish work. And so back to this, right, in terms of these zebrafish models, what have been found is that there have been promising findings on anti-inflammatory and neuroprotective compounds that have been found based on the work on these models. And for example, very sorry for that. So for example, corticosteroids, calcium channel blockers, and even certain antioxidants have shown recovery-promoting effects in zebrafish injury models before we want to maybe further test them on mammals. Of course, reproducibility and context specificity would remain the major concern, right? So, and also despite what we found is that just now I mentioned about the fact that zebrafish has these regenerative capabilities, right? Which would actually be something that sets them apart from rodents, is that although there are recovery in the functional integrity by 42 days post-injury in some other studies, it was found that up to 50% of axonal tracts that are located rostrally of the injury site remain actually unrepaired. So this incomplete regeneration is suggested to be due to deficiency in axonal myelination. So even at 1 year post-injury, in fact, the spinal cord tissue in the adult zebrafish, cannot fully regenerate. Okay, and so, cannot fully regenerate to its pre-injury levels. So that's something to consider. And in terms of the expression, right, what have been found, there are certain signaling pathways that have been found to be playing a role, whereby the sonic hedgehog signaling pathway was found to be upregulated following the stab injury model. And this SHH expression, right, it will activate the proliferation of radial glia cells, okay? And this is something that shows that it supports the anti-edematous and anti-inflammatory effect, and it can reduce seizure activity in these adult zebrafish as well, okay? And also, BDNF has been found to increase at 1 day post-injury, and it will gradually decline by the 15-day post-injury mark in the injured hemisphere. So these are talking about, for example, the stab injury model, and also certain other genes like GFAP as well as S100B will also be gradually increase. GFAP will be decreasing before increasing, whereas S100B will be gradually increasing in these models. So,
Sorry, Dr. Rozaida.
yes.
Yeah, okay, we are coming
We're
up to the
coming
wrap
to the
up.
conclusion. Yes. Okay. So basically, my short conclusion would be that the zebrafish gives us speed and scalability due to the relatively higher throughput, but we still need precision. There has been many pilot attempts for the student to be able to consistently inflict the injury in a very reproducible way. And careful randomization of these models are essential for meaningful data. So these are the key references, and I would like to show my acknowledgement. Thank you very much.
Thank you, Dr. Rozaida. That's this really cool talk on how we can fully utilize the zebrafish model. And we can take a couple of questions, I think there's just enough time for a couple of questions. We do have one question already in the chat from Ilman. And the question is, since zebrafish are social animals, does that qualitative change in behavior, is that qualitative change in behavior something you're investigating in this experiment?
Yes, in fact, the type of qualitative behavior, when we talk about behavior analysis, yes, there are the qualitative side of it. You have certain types of tests like the novel object recognition test, and then you also have the social behavior test as well. So definitely these are targeted to complement, as we said, what we claim as social animals.
Okay, thank you.
Okay.
And do we have any other questions?
But you said Ilman Shafi, we can talk further on this if you want to find out more about this.
Okay, thank you.
Okay, thank you very much.
And well, that very neatly ends our session from the biomedical science program. If anyone does have questions that just come up later, feel free to contact either Dr. Rozaida or myself. I'm the coordinator of Zebrafish Lab, and I'll be very happy to help anyone understand more about this lovely model. And now I pass the floor to Prof. Claire. Thank you, everybody.
Thank you.